Welcome back to the deep dive. Today, we are wading into territory that I think, well, most of us just take for granted.
It's almost a reflex, isn't it?
It is. We've all done it. You get that scratchy throat or maybe a sinus infection that just will not quit. And you go to the doctor expecting, maybe even, demanding a prescription.
You want the Z-Pack.
I want the Z-Pack. I want the amoxicillin. I just want the problem to be gone.
It is probably the most common medical transaction there is.
Right. But today, we are going to completely dismantle that transaction. We're not looking at antimicrobial therapy as just swallowing a pill to kill a bug.
No.
We're going to look at it through the lens of our source material as a high-stakes ecological intervention.
And that is the phrase from Goodman and Gilman that really changes everything. Ecological intervention. It's powerful.
And to guide us through this, we are synthesizing two absolute titans of pharmacology. We have Katzong and Trevor's Basic and Clinical Pharmacology, specifically Chapter 51. This is, effectively our field manual. It's the clinical roadmap of what to do and when.
The bedside guide.
Exactly. And then, providing the molecular backbone, we have Goodman and Gilman's The Pharmacological Basis of Therapeutics, Chapter 52.
And the distinction between these two, it's actually really important for how we should frame today's conversation. If Katzong is the what and the how.
The decision-making framework.
Right. For the clinician who is standing at the bedside, then Goodman and Gilman is the why. It's the deep dive into the mechanistic molecular reasoning and the systems-level biology.
So, okay, if Katzong tells you to drive the car at 60 miles per hour, Goodman and Gilman is explaining exactly how the combustion engine turns gasoline into torque.
That's a very fair analogy. And you need both. You absolutely need both. Because if you only know the how, you're just following a recipe.
And biology doesn't always follow the recipe.
Never. So you need the why to troubleshoot when things inevitably go wrong.
And they do go wrong. And that brings up a core tension that we have to address right up front. I think the average person, myself included before this reading, assumes the goal of antibiotics is simply kill the bacteria. Eradication.
Full stop.
Yeah. But the text suggests it's a lot more complicated.
Oh, it's so much more complicated.
Yeah.
The core tension is between that very idea of eradication, killing the pathogen, and another concept.
Restraint.
Restraint. As in holding back.
Holding back, exactly. Because every time you introduce an antimicrobial agent, whether it's an antibiotic, an antifungal, an antiviral, you are introducing a new chemical reality to a vast, complex ecosystem.
We're not just affecting the bug.
You are altering the host. That's the patient. You are attacking the pathogen, sure, but you are also fundamentally changing the wider microbial environment, your microbiome, in ways that are, often invisible until it's far too late.
So if you focus only on eradication,
you might use the biggest weapon you have every single time, and yeah, you might cure that patient today. But in the process, you wreck the patient's native gut biome, and even worse, you breed resistance that makes that very drug useless for everyone else tomorrow. So today is about finding that balance. We aren't just going to list drugs. We're going to build a conceptual map. We need to talk about selective toxicity.
how we poison the bad guys without poisoning ourselves.
Yes, we need to understand the pharmacophore.
Which is just a beautiful concept, the specific geometry of the drug molecule that does the work.
We'll also get into the math of warfare pharmacokinetics and pharmacodynamics. Why do some drugs need to be given constantly while others work best with one single massive dose?
And of course, the big one, the resistance arms race. The bacteria are adapting faster than we are discovering new drugs.
And finally, we'll anchor all of this in a really detailed clinical case study from Katzong that I think perfectly illustrates the difference between empiric guessing and definitive targeting.
A fantastic case.
So let's start with the basics, the magic bullet.
Yeah.
Historically, we credit Pasteur and Koch with the germ theory, this idea that specific invisible bugs cause specific diseases. That kicked off the race to find things that kill them. But Goodman and Gilman makes a really interesting point right off the bat that it stopped me. We shouldn't think of antibiotics as poisons.
No.
We should think of them as ligands.
Yeah.
Now, wait a minute. When I hear ligands, I'm thinking neuroscience. I'm thinking dopamine binding to a receptor in the brain or, adrenaline binding to a beta receptor.
Yeah.
That's a very sophisticated lock and key signaling mechanism.
Yeah.
Are you saying antibiotics work the same way?
Fundamentally, yes. That is the physics of it. I mean, in pharmacology, a ligand is just any molecule that binds to a specific receptor to trigger or to block a biological response.
Okay.
When we treat heart disease, we use drugs that bind to receptors in the heart. Antimicrobial therapy is the exact same physics. The antimicrobial molecule is the ligand. Its receptor just happens to be a protein inside the bacteria rather than a protein on one of your own cells.
So it's not like pouring bleach on a countertop where it just chemically burns everything it touches.
No, absolutely not. Bleach is a general protoplasmic poison. It destroys proteins indiscriminately. If you drank bleach, yeah, it would kill the bacteria in your stomach, but it would also dissolve your esophageal lining and, well, kill you.
That isn't medicine.
That's just destruction. Antibiotics are magic bullets, precisely because they are highly specific keys looking for highly specific locks.
And this leads to that term you mentioned that Ehrlich coined, the pharmacophore.
Paul Ehrlich, one of the fathers of chemotherapy. He introduced this concept to describe the specific part of the drug molecule.
The business end.
The business end, yes. The active chemical moiety that actually fits into the microbial receptor. Think of a key. The handle of the key lets you hold it and turn it. That might be the part of the drug that lets it dissolve in your blood.
Okay.
But the teeth of the key, that's the pharmacophore. That is the part that has to match the tumblers in the lock absolutely perfectly.
And this brings us right to the core concept of selective toxicity. This seems like the holy grail of drug design.
It is.
You want a pharmacophore that binds tightly to a receptor that is essential for the microbe, but, and here's the kicker, that receptor needs to be either completely absent in humans or at least significantly different.
Correct. That is the definition of a safe antibiotic. You want to target a biological pathway that the bacteria needs to survive, but that the human host, well, either doesn't use at all or uses in a very different way.
Give us the gold standard example of this. What's the easiest, most obvious target?
Oh, the classic one is the cell wall. Human cells are eukaryotic. We have cell membranes, these flexible fatty skins.
Right.
But we don't have cell walls. Bacteria, however, have these rigid armored outer shells made of peptidoglycan. They need them to keep from exploding under osmotic pressure.
So if you target the wall,
You are targeting a structure that simply does not exist in the human body. It's a perfect target.
and that's what penicillins do.
Exactly. If you have a drug like penicillin or a cephalosporin that inhibits the enzymes responsible for building that wall, it's like knocking out the steel girders of a skyscraper while it's being built.
It just collapses.
The bacteria literally lies. They explode.
Bruh.
But because human cells don't have that wall and don't have those enzymes, the drug just bounces right off of us. That is the definition of high selective toxicity.
Okay, that makes perfect sense for the cell wall. But what about when the targets are more similar? I know both bacteria and humans need to make proteins. We both have ribosomes.
Now that gets trickier. This is where we have to rely on evolutionary divergence. Both bacteria and humans use ribosomes to stitch amino acids into proteins, absolutely.
Right.
But over billions of years, those ribosomes have drifted apart in their structure.
So it's like a Mac versus a PC. They both do computing, but the software, the architecture, it's not compatible.
That's a decent analogy, yeah.
Yeah.
Bacterial ribosomes are composed of what we call 30S and 50S subunits. Human ribosomes are larger. They're made of 40S and 60S subunits. They have different shapes, different nooks and crannies, different binding sites.
So a drug can be designed to fit one and not the other.
Precisely. Drugs like tetracyclines or macrolides or aminoglycosides are designed to jam the gears of the bacterial 30S or 50S subunit specifically.
But they don't fit into the human ribosome at all.
They don't fit well, and that's a really crucial distinction. There is rarely 100% selectivity. At high enough concentrations or in certain tissues, there can be crossover.
And that's where side effects come from.
That's exactly where side effects come from.
Right.
For example, the mitochondria in your cells, the little power plants, they are thought to have evolved from ancient bacteria.
Right, the endosymbiotic theory.
Exactly. And their ribosomes actually look a lot more like bacterial ribosomes than human ones. So if you blast a patient with too much of a drug like linezolid or chloramphenicol, you can start inhibiting the protein synthesis in their own mitochondria.
And that causes toxicity.
That's where you get bone marrow suppression or lactic acidosis. It's off-target toxicity.
So the magic bullet is more like a magic-ish bullet. It hits the target most of the time, but there's a risk of collateral damage if you're not careful.
Precision is always relative. But compared to early treatments like arsenic or mercury, modern antibiotics are incredibly precise instruments.
Goodman and Gilman mentions some newer frontiers here, too.
Right.
We aren't just targeting walls and ribosomes anymore. It mentions an antisense antibiotic. That sounds like something from a sci-fi novel.
This is really the cutting edge, and it's absolutely fascinating. Traditional antibiotics target a protein, an enzyme, or a structural block. Antisense antibiotics target the information itself.
The mRNA.
The blueprint,
Yeah.
exactly. Before a protein can even be made, the DNA is transcribed into messenger RNA. Antisense molecules are short, engineered strands of nucleic acids that are complementary to a specific bacterial mRNA sequence.
So they just stick to it.
They bind to it, yeah. They lock it up in a double helix so the ribosome can't read it. It's like intercepting the construction orders before they even reach the foreman on the job site.
And because the sequence of letters, the A's, T's, C's, and G's, in bacterial DNA is specific to that bacteria.
You could, in theory, achieve extreme precision. You could design a drug that kills only a specific pathogenic strain of E. coli and leaves all the beneficial E. coli in the gut completely alone.
Wow.
We aren't fully there yet in the clinic, but that is the absolute dream of selective toxicity.
Okay, so we've designed this perfect ligand. We've got the key for the lock. We swallow the pill. Now comes the hard part.
The journey.
The journey. It doesn't matter how perfect your missile is if it can't reach the target. This is what the texts call the battlefield, or pharmacokinetics.
Getting the drug to the site of infection. It sounds so trivial, but it is the cause of treatment failure in a huge percentage of cases.
I think people have this mental model that the body is just a bucket. You pour the drug in, it mixes around, and it touches everything equally. But Goodman and Gilman spends a lot of time explaining compartments.
The body is definitely not a bucket. It's a maze. A maze of distinct compartments, each with its own barriers, its own pH, its own blood flow. And before we even talk about where the drug goes, we have to talk about the relationship between how much drug is there and how much killing actually happens.
The dose-response relationship?
Exactly.
Yeah.
This is the Hill curve, or the sigmoid Emax model.
Okay, let's break that down. It's an S-shaped curve.
Right. If you graph the concentration of the drug on the bottom axis and the killing effect on the vertical axis, you don't get a straight line. It's not double the drug, double the kill. It's an S shape.
So walk us through that S-curve from left to right.
Okay, at the bottom left, you have very low concentration. The curve is flat, zero effect. You're giving the drug, but it's below the minimum inhibitory concentration, the MIC. The bacteria are, frankly, laughing at you.
Nothing is happening.
Nothing. Then you hit a certain point, a threshold, and the curve shoots up steeply. This is the sweet spot.
That steep part is called the slope. Why does the slope of that line matter to a doctor at the bedside?
The slope, or the Hill factor, tells you how sensitive the response is to a change in concentration. If a drug has a very steep slope, a tiny increase in concentration gives you a massive increase in killing.
Which sounds good.
It is good, but the reverse is also true. A tiny drop in concentration can cause the effect to crash back down to zero.
So if I'm a patient and I miss a dose by, say, 2 hours and the drug level dips just a little bit.
If it's a steep slope drug, your therapy might fail completely during that 2-hour window.
Yeah.
The bacteria get a chance to recover. If it's a shallow slope drug, you have more wiggle room, more forgiveness. So knowing the shape of that curve is crucial for dosing schedules.
And then at the very top, the curve flattens out again.
That's Emax,
Yeah.
the ceiling. This is the maximum killing capacity of that particular drug mechanism. Once you hit Emax, you can pour in 10 times more drug, 100 times more, and you won't kill a single additional bacteria.
You'll just cause toxicity.
You will just poison the patient. Understanding Emax is what prevents us from overdosing in a futile attempt to get a better result.
And there's one more variable on this curve that we see all the time in lab reports, the IC50.
The inhibitory concentration 50. It's simply the concentration you need to get 50% of that maximum effect. It's a measure of potency.
So a lower IC50 is better.
Much better. It means you need less drug to do the job. And when we talk about resistance later, this is what changes. The bacteria mutate, and suddenly that whole S-curve shifts to the right. The IC50 goes up.
Yeah.
You need more and more drug to get the same result until eventually the dose required is toxic to the patient.
The therapy becomes impossible.
Right.
Okay, so we have this math in our heads. We need to get the concentration up that steep part of the slope. But there are barriers, walls, gates. Let's talk about the central nervous system, the brain. The blood-brain barrier is legendary for keeping things out.
It has to be. The brain is the command center. It can't have random toxins from your lunch floating around your neurons. So the capillaries in the brain have these incredibly tight junctions. They're basically welded shut. To get in, a drug usually needs to be lipophilic. Right. It needs to be able to dissolve right through the fatty membranes of the cells. If a drug is hydrophilic water-loving, charged, polar, it just bounces off. It can't get in. Penicillin's a great example. It's polar. Normally, if you take penicillin, the level in your brain is almost zero.
But, and this is the part that just blew my mind in the reading, We treat bacterial meningitis with high-dose penicillin. How is that possible if it can't get in?
It's one of the great and frankly terrifying paradoxes of infectious disease. When a patient has meningitis, the meninges, the lining of the brain, are acutely inflamed.
Yeah.
And what is inflammation? It's swelling, redness, and crucially, the loosening of vascular barriers to let white blood cells get in.
So the infection itself opens the gates for the cure.
It opens the gates, exactly. The tight junctions loosen. The fortress walls develop cracks. So when the patient is at their absolute sickest, when they're comatose, the barrier is leaky. You give a massive dose of penicillin G intravenously, and because the barrier is compromised, it floods into the cerebrospinal fluid and it starts to work.
But here's the devastating catch. As the medicine starts to work,
The patient gets better, the inflammation subsides, the swelling goes down, and the tight
you
junctions
close up again.
zip themselves back up.
So by curing the patient, you are literally shutting the door on the cure.
Precisely. As they heal, the drug delivery to the brain drops precipitously. This is a huge, huge trap for the clinician.
Yep.
You see the patient improving, their fever is down, their mental status is clearing. You might be tempted to lower the dose or switch to a less aggressive drug.
Right, makes sense.
But if you do that too early, while the barrier is closing but a few stubborn bacteria are still alive, you get a catastrophic relapse. You have effectively locked the remaining bacteria inside a fortress where the drug can no longer reach them.
That is an incredibly tricky dynamic to manage. It's like the body is sabotaging the cleanup operation.
It's just physiology doing its job, but it complicates therapy enormously. And we see similar issues in other sanctuary sites. The eye is another one. Drugs penetrate the eye very poorly, which is why we often have to inject antibiotics directly into the vitreous humor with a needle.
Ouch.
Yeah, not pleasant. Or the prostate, which is notoriously difficult to penetrate. Or the pericardium around the heart. Goodman and Gilman cites a study on tuberculous pericarditis. They looked at rifampin levels, gray levels in the blood, but inside the pericardial sac, only 20% of the plasma level.
And why was that? Was it being pumped out?
Active clearance mechanisms. The body has pumps like P-glycoprotein that are designed to recognize foreign molecules and actively throw them out of sensitive tissues. So even if the drug gets in, the body is bailing it out like a sinking boat.
So if you're treating that patient with a combination of drugs, say rifampin and isoniazid, and only the isoniazid is getting good penetration into that specific tissue,
Then inside that pericardial sac, you're effectively using monotherapy.
which is a recipe for breeding resistance.
Exactly. You think you're using a cocktail to prevent resistance, but in that one pocket, the bug is only seeing one drug. That is how you breed a resistant mutant in a single compartment of the body, which can then spill out and reinfect the whole system.
Speaking of pockets of resistance, we have to talk about the ultimate fortress, the biofilm. This isn't just a barrier, this is a whole colony.
This is the nightmare scenario for modern medicine, especially with implants. Artificial heart valves, prosthetic hips, IV catheters. We tend to study bacteria as planktonic, as these free-floating swimmers in a test tube.
But that's not how they live in the body.
No. In the body, they like to settle down. They attach to a surface and they secrete this exopolymer matrix,
Just slime, basically.
a very complex, structured slime city. And this matrix does two critical things. First, it's a physical and chemical shield. The matrix is often negatively charged. So if you throw a positively charged antibiotic like an aminoglycoside at it, the drug just gets bound up in the slime grid. It never even touches the bacteria deep inside.
So it's like a chemical trap.
It is. And the second thing, metabolic dormancy. Antibiotics generally work by sabotaging active machinery, building a wall, making DNA, making proteins. But deep inside a biofilm, the bacteria go into a sort of hibernation state. They aren't building walls, they aren't replicating, they are just existing.
So the antibiotic has no target.
It has nothing to attack. You can have a drug that kills millions of planktonic bacteria in seconds. But against that same bacteria in a biofilm, it does absolutely nothing. This is why standard susceptibility testing, the MIC numbers you get from the lab, are totally useless here. They're testing the planktonic form.
So what do if you have a patient with an infected artificial hip and a massive biofilm on it?
Often, there is no chemical solution.
Yeah.
You have to operate, you have to physically go in, rip the hardware out, debride the area, wash it out, and start over. It is a fundamental failure of chemistry.
That is a very sobering reality check. Okay, so moving on. We've managed to navigate the barriers. We've reached the bug. Now we need to know the rules of engagement. This is section 3 in our outline. PK-PD integration.
This is where we put the motion of the drug, the PK, and the effect of the drug, the PD, together. And it turns out how much drug you give and how often depends entirely on how that specific drug kills that specific bug.
Goodman and Gilman breaks this down into three main drivers of efficacy. I want to use an analogy here, if that's okay. Let's imagine the bacteria is a door we are trying to force open, or maybe keep shut, depending on how you look at it.
Okay, I like analogies. Go on.
The first driver is time-dependent killing. The formula is T is greater than MIC.
Right. For these drugs, the peak concentration doesn't really matter that much.
Yeah.
As long as you are above that minimum inhibitory level, you are winning. Going 10 times higher doesn't help. What matters is time.
So in my door analogy, this is like holding a door shut against a zombie. You don't need to be Superman. You don't need to push with 5,000 pounds of force. You just need to apply enough pressure to keep the latch clicked.
Okay.
But, and this is the key, you cannot walk away. If you stop leaning on the door for 10 minutes, the zombies get out.
That is a perfect description of beta-lactams, the penicillins and cephalosporins. They work by inhibiting cell wall synthesis. The bacteria are only vulnerable when they are actively trying to build that wall, which they do continuously.
So you have to be there all the time.
All the time. If the drug level drops below the MIC for even a few hours, the bacteria rebuild, they recover, and they start replicating again. You lose control.
So what does that mean for dosing? You can't just give one giant pill on Monday and come back on Friday.
Absolutely not. You need frequent dosing, every 4 hours, every 6 hours. Or what we're doing more and more in hospitals is using continuous infusions. We hook the patient up to an IV pump that drips the drug in at a slow, steady rate, ensuring the level never drops below the MIC.
You're leaning on that door 24/7.
We are leaning on that door 24/7.
Okay, second driver, concentration-dependent killing. The formula here is Cmax divided by MIC.
This is a totally different beast.
Yeah.
Here, the peak matters immensely. The higher the peak concentration, the more bacteria you kill and the faster you kill them.
So this isn't leaning on the door. This is a SWAT team with a battering ram.
Yes.
You want one massive explosive hit to shatter the door completely off its hinges.
That's it. Drugs like the aminoglycosides, like gentamicin, and the fluoroquinolones, like ciprofloxacin, work this way. You want to achieve a peak concentration that is maybe 10 or 12 times the MIC.
Yeah.
A massive, overwhelming spike.
And because you shattered the door, you don't need to stand there and guard the empty frame afterwards.
Exactly. And this leads to a fascinating phenomenon called the post-antibiotic effect, or PAE. Even after the drug is excreted by the kidneys and the levels in the blood drop back to zero, the bacteria don't grow back immediately.
They're stunned.
They're stunned. They have to repair non-lethal damage to their ribosomes. They have to resynthesize enzymes. They're knocked out.
It's like a boxer who's on the canvas. The punch is over, but he isn't getting back up for a while.
Right. And this is clinically huge for drugs like aminoglycosides, which can be quite toxic to the kidneys and inner ear. Because of the PAE, we can give one huge dose a day, get that massive killing spike, and then let the drug wash completely out of the patient's body for the next 18 hours.
So the patient's kidneys get a break.
The patient's kidneys get a break to recover, but the bacteria stay suppressed. It's a win-win that lets us use a powerful but dangerous drug safely.
That's fascinating. So dosing frequency isn't just about convenience, it's about exploiting the specific weakness of the bug while sparing the organs of the host.
Everything in pharmacology is a trade-off.
Yeah.
It's always about maximizing efficacy while minimizing toxicity.
There's a third category too, right? Total exposure.
Yes, the AUC over MIC drugs. Here, it's not the time or the peak that matters so much as the cumulative amount of drug the bacteria sees over a 24-hour period. It's the total area under the concentration curve. Drugs like vancomycin or daptomycin fall into this category.
Okay, so there's one more critical distinction from Katzum we need to hit before we talk about resistance. Bacteriostatic versus bactericidal.
Static means stationary. The drug inhibits growth. It freezes the bugs in place, but it doesn't actually kill them. It relies on your own immune system, your white blood cells, to come in and mop up to eat the frozen bacteria.
Okay.
Cidal means the drug kills the organism outright. It does the job itself.
And for most people with a healthy immune system, static is perfectly fine, right? If I get a run-of-the-mill sinus infection, a static drug like doxycycline works great.
For a healthy immunocompetent person, yes. But, and this is a life or death distinction, what if the patient is neutropenic? What if they have no functional white blood cells because of chemotherapy?
Then there's no cleanup crew.
There is no cleanup crew.
Yep.
Or what if the infection is in a place the immune system can't easily reach, like on the heart valves and endocarditis, or in the cerebrospinal fluid and meningitis? Those are immune-privileged sites.
So there are no police to come and arrest the frozen criminals you've captured.
Exactly. In those specific cases, endocarditis, meningitis, and in profoundly neutropenic patients, you must use a bactericidal drug.
Ah.
If you use a static drug, you are just pausing the infection. As soon as you stop the drug, it will come roaring back. You cannot rely on host defenses that don't exist or can't get to the fight,
It effectively becomes a chronic management issue rather than a cure.
which is a complete disaster for a condition like bacterial meningitis.
Okay, let's turn the page to the darker side of the story. Section 4. The enemy adapts, resistance ecology.
I like that you call it ecology because it isn't just bad luck. It is evolution in fast forward. Goodman and Gilman puts it starkly. Antibiotics are an existential threat to bacteria.
Right.
And Evolution 101 says if you threaten a species with extinction, the survivors will be the ones who, by random chance, developed a mutation that let them survive. We are applying massive, relentless selection pressure.
But it feels like they adapt so fast, faster than normal evolution should allow.
Well, for one, they reproduce every 20 minutes, so they get a lot of rolls of the dice. But some bacteria are actually, in a way, cheating. They have what we call hypermutable phenotypes.
Mutation factories.
That's a great way to put it, yes.
Oh.
Take the M. tuberculosis Beijing genotype, for example. It's a particularly nasty strain of TB. It has known defects in its own DNA repair systems. It makes mistakes on purpose during replication.
Why would it do that?
Now, 99.9% of those mistakes might be useless or even fatal to the bacteria.
Yep.
But because it's rolling the evolutionary dice billions and billions of times, it hits the jackpot, a useful resistance mutation, much, much faster than a stable bacteria ever would.
It's brute forcing the evolutionary law.
Exactly. It's a high-risk, high-reward strategy that pays off under pressure.
So how do they do it? What are the actual mechanics of resistance, if we go back to our lock and key analogy?
We can categorize them into a few main strategies. First and simplest is reduced entry. Just shut the door.
Lock them out.
Lock them out. Bacteria like Pseudomonas have these protein channels called porins in their outer membrane that let nutrients and also drugs get inside. They can just delete the gene for that porin or mutate it so it's closed. If the drug can't get in, it can't work.
Simple, very effective.
Second strategy is efflux pumps. We mentioned P-glycoprotein in humans, but bacteria are masters of this. They have pumps that act like little molecular sump pumps. As soon as the antibiotic molecule enters the cell, this pump, often powered by a proton gradient, or ATP, grabs the molecule and spits it right back out.
So it's like trying to bail out a leaking boat, but the bacteria is the one bailing out our drug.
Precisely. The malaria parasite, P. falciparum, is famous for this. The drug chloroquine works by accumulating to toxic levels in the parasite's digestive vacuole. The parasite evolved a specific transporter, BFMDR1, whose whole job is to pump the chloroquine out as fast as it comes in. We lost a miracle drug to a simple pump.
Wow. Okay, third strategy.
Enzymatic destruction. This is the most aggressive approach.
Yeah.
The bacteria don't just block the drug, they build a weapon to destroy it. They produce enzymes like the infamous beta-lactamases that drift around and physically chew up the antibiotic molecule before it can even reach its target.
And break the chemical bond.
They cleave the beta-lactam ring. It's like shooting down the missile before it hits the building.
And fourth.
Altered target. This is the most subtle. Change the lock. This is the mechanism behind MRSA, methicillin-resistant Staphylococcus aureus.
Right.
Methicillin and other beta-lactams work by targeting a specific penicillin-binding protein, or PBP. MRSA acquired a new gene called mecha that codes for a totally new PBP called PBP2A, which has a slightly different shape.
So the drug doesn't fit anymore.
The drug arrives, it looks for the lock, but the lock has changed. It can't bind. The bacteria just keeps building its cell wall, completely ignoring the drug that's all around it.
Now, if these mutations stayed in one parent bacteria and just got passed down to its children, that would be bad enough. But bacteria have, well, they have what amounts to sex, don't they? Horizontal gene transfer.
It is the internet of bacteria. They file share. Vertical transfer is parent to child. That's slow. Horizontal transfer is neighbor to neighbor, even across completely different species.
It's like if I learned how to play the piano and I could just touch your arm and suddenly you'd know how to play the piano too. And not just you, but the dog and the cat in the room.
That is a terrifyingly accurate analogy. There are three main ways they do this. Conjugation is the sex part. Two bacteria physically connect with a thin tube called a pilus. One sends a copy of a plasmid, a small mobile ring of DNA, over to the other. These plasmids are notorious for carrying resistance genes.
So they swap superpower cards.
They do. Then there is transduction. This involves viruses, called bacteriophages, that infect bacteria. Sometimes, when a new virus is being assembled, it accidentally grabs a piece of the bacterial DNA instead of its own viral DNA.
And it injects it into the next bug it infects.
It delivers it like a little syringe. And transformation is the simplest of all. That's just scavenging. Bacteria can literally pick up naked DNA that's floating around in the environment from a dead, leased neighbor. Oh, look, a gene for penicillin resistance. I'll just take that.
And the most dangerous part of this is that these genes often travel in packs, right?
Yes, on things called integrons and transposons. These are jumping genes. They can collect multiple resistance cassettes. One for penicillin, one for tetracycline, one for sulfa drugs, all into one single mobile plasmid. One transfer event and a harmless bug can become a multi-drug resistant superbug instantly.
The ultimate example of this, and the stark warning Goodman and Gilman gives us, is the colistin story. This one really kept me up at night.
The MCR1 gene. It is a tragedy of our own making, a cautionary tale.
Yeah.
Colistin is an old antibiotic. It's a harsh drug. It acts like a detergent on cell membranes.
So it's not very selective.
No, it's nephrotoxic, meaning it damages the kidneys, and it's neurotoxic. We stopped using it in humans decades ago because we had much better, safer options. It was put on the shelf as a break glass in case of absolute emergency drug.
A drug of last resort. But we didn't stop using it everywhere.
No, we started using it by the ton in agriculture, specifically in pig farming in China and parts of Europe.
To treat sick pigs.
No, largely for prophylaxis and as a growth promoter, to fatten them up cheaper and faster. So we soaked the global pig population in our last resort antibiotic. And naturally, a resistance mutation emerged, Panchami in the 5-1. But here's the terrifying twist. It wasn't on the chromosome. It was on a plasmid. It was mobile.
So the bacteria in the pigs get resistant. We eat the pork.
The resistant bacteria enter the human gut. The plasmid then jumps from the harmless pig E. coli to human pathogens that are already living in our own gut microbiome. And suddenly, a few years ago, we started seeing patients in ICUs with infections resistant to colistin, our last resort drug.
And these patients had never taken colistin.
Never. They had no history of it. But the resistance was bred in a barn, possibly halfway across the world, and shipped to them on their food.
It really drives home the point that you cannot separate clinical medicine from agricultural ecology. It is one single, interconnected microbial world.
We're all swimming in the same gene pool. The antibiotic poured into a trough in one country eventually, inevitably, impacts the ICU in another.
Okay, that's the doom and gloom. We understand the enemy, we understand our weapons. Now, let's talk strategy. How do we actually treat patients without causing a global apocalypse? This is the clinical application section.
We have to move from the molecular to the bedside. And to do this, I want to really dig into a case study from CatZone. It's a classic scenario that every medical student and resident needs to understand inside and out.
Okay, set the scene for us.
Okay, you're the resident on call in the emergency department. 65-year-old man is brought in. He's confused, he's shaking with chills, his blood pressure is soft, meaning it's starting to drop. He has a high fever.
Sounds like sepsis.
Classic sepsis picture. His wife tells you that he had a urinary tract infection, a UTI, about 2 weeks ago.
Okay, UTI, common stuff. Was he treated for it?
Yes. He went to an urgent care clinic. They gave him a prescription for ciprofloxacin for 3 days. He took it, he felt better, but now he's back, and he looks like he's dying. Oh, and one other minor detail the wife mentions. They just got back from a month-long trip to India 3 weeks ago.
India. Okay, in this context, why does that specific geography matter so much?
Geography is destiny in infectious disease. India, unfortunately, has a very high prevalence of highly resistant bacteria, particularly extended spectrum beta-lactamase or ESBL producing organisms, and also very high rates of fluoroquinolone resistance.
So let's break down what likely happened here. He took the Cipro, he got better. Why is he crashing now?
Think about the ecology of his bladder. He had an infection. It was probably a mixed population of bacteria. Most of them were susceptible to Cipro. But maybe a tiny fraction, let's say 0.01%, were resistant, perhaps a strain he picked up during that travel.
Okay.
He takes a Cipro.
The Cipro kills off the 99.9% that are susceptible.
Right. And his symptoms go away. He feels cured. But biologically, what did we just do? We cleared the entire playing field. We killed all the competitors. We left that tiny 0.01% of resistant superbugs alone with plenty of nutrients and space to grow.
So they just took over.
They roared back. And this time, the entire population is 100% resistant. This is the relapse. And now it's not just a UTI. He has urosepsis. The bacteria have spilled into his bloodstream.
So you're standing there in the ER. He's crashing. You need to start antibiotics right now. You can't wait 2 days for a lab test. What do you do?
This is the empiric phase of therapy, the best guess. and the stakes are life and death. Every hour you delay appropriate antibiotics and septic shock increases the risk of mortality by about 8%. You cannot afford to be wrong.
So you're not giving Cipro again?
Absolutely not. You have to assume you already failed with Cipro. You have to assume this is a highly resistant bug. You have to escalate. You bring out the big guns.
What does that mean?
You might start at carbapenem, like meropenem, or perhaps piperacillin tazobactam. You need a very broad-spectrum drug that covers the worst-case scenario, given his history. You cover everything.
Broad spectrum, kill everything, but that's just the first step.
Yes. That is day 1. It's a necessary evil. Now flash forward to day 3. The patient is stable, his fever is gone, his blood pressure is up, and the lab report comes back.
This is the definitive phase.
This is the pivot point.
Yeah.
The culture from his blood grows E. coli. The sensitivities say resistant to Cipro, resistant to ampicillin, but sensitive to trimethoprim sulfamethoxyls, also known as Bactrim.
Bactrim, the cheap old narrow-spectrum drug.
The cheap old drug. Now, the doctor has a choice. The patient is doing great on the meropenem, the Ferrari of antibiotics. It feels safe and easy to just keep it going. Don't rock the boat, right?
But that's the wrong choice.
That is the ecologically disastrous choice. If you keep using that broad-spectrum meropenem, you are applying massive selection pressure to his gut flora. You are breeding the next nightmare, a carbapenem-resistant organism. Good antimicrobial stewardship demands that you de-escalate.
You take him off the Ferrari.
You stop the Ferrari, you switch to the Bactrim. It's narrow, it's targeted, it works, and it spares the rest of his microbiome from collateral damage.
That switch from broad empiric to narrow definitive seems to be the hardest thing for humans to do psychologically. We like the security blanket of the big, powerful drug.
It is a huge challenge. But good, responsible medicine requires overcoming that fear and trusting the data.
One last strategic question, combination therapy. We've established that monotherapy one drug is usually preferred to keep toxicity and cost down. But are there times when 1 + 1 = 10?
Yes. The concept of synergy. The classic example is enterococcal endocarditis, a nasty infection of the heart valve by a tough little bug called Enterococcus.
Why is it so tough to treat?
It's not particularly virulent, but it's incredibly stubborn. If you give penicillin alone, it only inhibits the bug. It's bacteriostatic, it doesn't kill it. If you give an aminoglycoside like gentamicin alone, it can't even get into the bacteria because the cell wall is too thick.
So both drugs fail on their own.
They're both ineffective individually. But if you give them together, the penicillin punches holes in the cell wall. It doesn't kill the bug, but it weakens the defenses. It opens the door.
And the gentamicin just walks right through that open door.
The gentamicin walks through the holes made by the penicillin. It gets inside, it reaches the ribosome, and it kills the bacteria.
Yeah.
You achieve a bactericidal cure from 2 drugs that fail on their own. It's a beautiful, elegant molecular partnership.
And the other big reason for combinations is to prevent resistance from emerging, right? Like in TB or HIV treatment.
That's purely a numbers game. In tuberculosis, the rate of a spontaneous mutation that confers resistance to, say, isoniazid is maybe 1 in a million bacteria. That's 10 to the -6.
Huh?
But a patient with a big cavity in their lung might have a billion bacteria in there. So if you do the math, there are already 1,000 resistant mutants in there before you've even given the first dose.
So if you use just isoniazid, those 1,000 mutants survive and take over the whole infection.
Exactly. But the odds of a single bacterium having a random mutation to isoniazid AN, a random mutation to rifampin at the same time, that's 10 to the -6 times 10 to the -6, that's 10 to the -12, 1 in a trillion.
That's practically impossible.
It is. So we use 3 or even 4 drugs at once. We make the mathematical probability of a single organism being resistant to everything at the start effectively 0. It's a statistical trap we build for the bacteria.
We have covered such a huge arc here, from a single ligand binding to a single receptor, to the S-shaped math of the Hill curve, to the global spread of resistance genes from pig farms, all the way to these life and death decisions in the ER.
It is a massive and humbling topic.
If we pull back to the 30,000-foot view, what is the one thing you want our listener to change about their mindset when they think about antibiotics?
I think we need to retire the war metaphors. We spent the entire 20th century obsessed with the magic bullet and fighting the war on germs. We thought we could conquer nature. We thought we could eradicate infection from the face of the earth.
And nature pushed back.
Nature always pushes back. The 21st century has to be about restraint. It has to be about stewardship. We have to realize that every time we use these drugs, we are borrowing from the future. We are extracting efficacy from a finite, precious resource.
So collateral damage isn't just a side effect, it's actually the main effect of overuse.
It is. We treat the infection in the lung, but in the process, we ravage the microbiome in the gut. We select for C. difficile, we breed resistance that spreads to the whole community.
So the provocative thought to leave you with is this. Maybe treating an infection is as much about what you don't do as what you do. It's about knowing when to hold your fire.
Stewardship isn't just about saving money for the hospital. It's about saving the molecules themselves so they still work when our children and grandchildren really need them.
If you want to see the charts, the structures, the detailed dosing tables, go to Katsung chapter 51 and Goodman and Gilman chapter 52. Use this deep dive as your conceptual map, but you absolutely have to walk the territory yourself.
Read the sources.
Yeah.
The details truly matter.
Thanks for listening to the deep dive. We'll see you next time.